move on to introduction. Nasopharyngeal carcinoma, or also known as NPC, is the mutation occurs in the epithelium of the nasopharynx. It is also denoted as the 5th highest cancer among Malaysians. Moving on to p73 gene, the gene has been categorized as one of the p53 families. It has been known to promote cancer progression by catalyzing anti-oxidizing reaction. It also has been known to inhibit senescence. Let's go to problem statement. The problem statement of this experiment is p73 has similar homology and structure to the p53, which is known as the tumor suppressor gene. This gene, by studying this research, by conducting this research, we can actually provide valuable insight into the disease and the possible treatments. Currently, there are insufficient studies between the gene expression analysis of the p73 gene with nasopharyngeal carcinoma. The objective of this experiment are two. Firstly is to detect the mRNA levels, mRNA presence of p73 gene in the TW04 NPC cell line and HK1 NPC cell line. Second is to distinguish the levels of mRNA between the TW04 NPC cell line and the HK1 NPC cell line. Let's go to literature review. What is NPC? NPC is the uncontrolled mutation that occurs in the epithelial cells located in the nasopharynx. The cell line that I'm using firstly is the TW04. It is a cancerous cell line, has been derived from a 36-year-old Taiwanese NPC patient. It is known as an undifferentiated squamous cell carcinoma. It tends to adhere to each other and the surface, and there is no presence of Epstein-Barr virus. Next is the HK1. This is also a cancerous cell line, has been derived from a 59-year-old Chinese NPC patient, and this is known as a well-differentiated squamous cell carcinoma. It tends to have a nuclear karyotype with structural abnormalities, and there is no presence of Epstein-Barr virus. What is the p53 gene family? As you can see in the figure that I have attached, you can see that p73 has similar structure to p53, and the gene itself is located in the chromosomal region 1p36.32. It has been known to be responsible for the regulation of cell cycle and apoptosis. It tends to be tumor suppressive and proto-oncogenic, and it has been known to activate p53 target and regulate tumor suppressive abilities. The methodology of this experiment firstly is the NPC cell culture, which I have been sourced from the animal tissue culture lab by one of the postgraduate students. Secondly is the RNA extraction from both of the cell line. Thirdly is the RNA quality checking using nanodrop. Fourth is the RNA validation checking for both of the NPC extraction using agar gel electrophoresis. Fifth is the primer sequence, which are p73 and GAP-PDH primer. Sixth is the primer PCR gradient for p73 primer has been done. Seventh is the synthesis of the cDNA, which I converted the RNA into cDNA. Eight is the polymerase chain reaction step, where the sample will undergo PCR. Next is the PCR product that has been obtained will be sent to ethical scientific to be sequenced. Moving on to the results of my experiment. Firstly is the PCR product visualization using agar gel electrophoresis. As you can see, both of the cell line, the sample does not have any prominent band shown. However, for TW04 NPC cell line, the sample too has a slight smearing. Despite the smear, I cannot say that that is a good result, because it is not the first try that I get that, so I had to proceed with that smearing, and accept it is. However, for my external control, by using gap PDH as the primer, I obtain a band which has the range of 200 to 300 base pair, which I have the product target size is approximately 252 base pair. Next is the PCR product sequencing. For TW04 NPC PCR product, the results are inconsistent, and the reverse primer for sample 1 and sample 2 does not have any sequence nucleotide. So I cannot, there is no detection of the sequence for the reverse primer. However, for HK1 NPC PCR product, for forward primer, there is no sequence nucleotide detected, but for the rest of the sample, for forward reverse of sample 1 and sample 2 for the reverse, there is a sequence nucleotide detected. However, the results are inconsistent and the percent of identities varies. These results are not accurate. Thus, I would like to talk about my discussion. Incorrect sequence size has been obtained because, as you can see, from the sequence size that I have obtained, it varies than the target product size, which is the p73. The target product size that I want is actually 994 base pair. However, for TW04, the sequence size that I obtained for both of the forward primer is 126 base pair. However, for HK1, the sequence size that I obtained is bigger than the target product size that I want. Thus, I can say that this is the result of non-specific amplification caused by non-specific primer. The reaction that occurs throughout the PCR reaction is cross-reactivity and false amplification. This allows the results of non-specific PCR product that I have obtained in the sequence analysis. And as the primer binds inaccurately to the target sequence obtained. Conclusion and recommendation. P73 mRNA expression could not be calculated as the primer binds to the wrong target sequence. Specific primer is needed in future study to allow the study of gene expression levels of P73 gene in NPC to be done. These are my references, and thank you.
Thank you, presenter. Now it is Q&A session. The examiner is Dr. Hong.
Thank you. Dr. Aikan. Thank you for the presentation. Can you go to the conclusion? You say you are saying that the primer binds the wrong target sequence. What do you mean? Can you explain to me?
The primer binds to the target sequence, meaning that the primer itself is non-specific, meaning that it binds randomly to the target sequence that I want, because in the sequence analysis that I obtained, I'm supposed to get the P73 gene. However, the sequence analysis shows random sequence. Thus, I can say that.
So that means if the binding is successful, so that means you are expecting to get 994 base pair.
Yes.
Okay. So this primer, is it designed by you yourself?
Unfortunately, the primer is the primer that I obtained inside the lab. Previously, at first, we had to do primer identification because in here, right? And where is it? In here, right? As you can see, the GC percentage for the reverse primer is very high. So I can when we do primer identification, we have to choose the GC percentage that is lower than 50% it can, which is why the primer that I use is the primer that I obtained, is not the primer that I designed.
So the means this primer is not designed by you?
No.
Oh, okay. So is it free? Does this primer be used before?
Yes. It has been designed by one of the postgrad students.
Can you go to the carousel photo showing the
Yes.
External means
Positive
okay,
control.
so that one is election G. And then the one is blue color is sample 1 and 2, right?
Yes.
So just now you are mentioning that the sample 2, the one that you mentioned, the one is for the P73 and PC cell line, right? So you are claiming that sample 2 actually is slightly better compared to other sample, am I right?
In terms of the visualization,
Okay.
yeah.
So do you think that what is the desired product?
No,
No, right?
because I'm supposed to get higher than that, but the range is less than 300.
Okay. So apa itu? So the one is what?
I think contaminant,
Is primer time.
primer dimer contaminant.
So what do you think? Do you agree that one is actually is primer time?
I cannot say that because I did not sequence my gap PDH. If I sequence my gap PDH, I can confirm that it is a contamination that occurred. But because I did not sequence my gap PDH, I'm not sure whether the gap PDH itself I get the right sequence.
I think your external control is okay, perfect.
Okay.
Nothing wrong. Now is your sample. Yeah.
Whether the sample have contaminants.
I think the
Is that-
sample is not going to matter.
It's just the primer is not specific.
It's not specific, but you just need to spend, and I think you have to put in extra effort. Okay, to do more optimization. Because that one is primer time well. Yeah. I think the quality of your RNA, I think is okay.
Yeah.
Acceptable use effort. It's just that maybe you need to put in more effort, with extra amount to get a better PCR result. Yeah. At least you got primer timer.
Because
That one is not contamination.
because I did learn from a postgrad student that maybe it's because my primer is it's not diluted. I did try to dilute it by using 90 microliter of nucleus-free water and 10 microliter of the primer itself. However, the result is still the same.
Okay. But do you look at the annealing temperature?
The annealing temperature
Do you optimize the annealing temperature?
by using the PCR
Gradient.
gradient. This one, I optimized the PCR annealing temperature of the primer.
Do you repeat this experiment?
I did not because yeah. Actually, there's a lot of time, but this one is the best result that I can get.
Actually, you can try different range of temperature. So, the sequencing. I'm just wondering, this one, that means you send your PCR product for sequencing. Which one?
The p73 primer.
That means the one is the include the primer timer, right?
Uh-uh.
So you send that one for sequencing.
Yes.